And for our last speaker for our breakout session 2, I'd like to introduce Dr. A. Malaswara Reddy. He is the head of analytical R&D at Dr. Reddy's laboratories. His title is Analytical Methods to Support Generic Drug Bioequivalence. And as our prior sessions, stay tuned for a in-depth panel discussion after his talk.
Hi, this is Maleshwar Reddy from Dr. Reddy's India. So today I'm going to talk about two topics. First one is complex product characterization, which are the most useful techniques for the characterization. The second section deals with rationale for sampling and batch-to-batch variability for various types of complex products. The first section of complex products, I have taken liposomal characterization. So in this, I have listed down here the several key characteristics and their most useful techniques. The first one is liposomal size distribution, wherein G-preciser can be used. Morphology, lamellarity, and bilayer thickness. The most useful technique is cryo-TEM. Lipid bilayer transition can be analyzed using nano-DSE. The physical state of active substance inside the liposome. In some liposomes, the drug is inside the liposomes, and some liposomes it is within the bilayer, so it can be analyzed using Sachs analysis. The free and encapsulated drug, we can use size exclusion technique followed by an HPLC. This is a very standard technique. Next one is in vitro drug release, which is important to understand our drug release characteristics in the human body. So again, the same techniques of size exclusion can be done, and ultracentrifugation also can be used as additional orthogonal technique. Internal pH is another important characteristics which can be easily assessed using ultrafiltration by excluding the external environment. The next important thing is a drug and lipid impurities, which are analyzed using LCMS and detectors like RI, LSD, and CAD, where the nanochromophoric impurities also can be characterized. The next set of products is characterization of long-acting injectables, which are typically polymer-based. So microsphere products all comes into this category. So in this kind of products, characterization of input polymer as well as the end product characterization is important. So when you start with input polymer characterization, so here I have listed the various kind of characteristics and most useful technique. The polymer composition is assessed using a proton NMR, where the monomer ratios are assessed. Molecular weight is assessed using GPC-RI or GPC moles, and degree of benching using moles, RI, and viscosity using a triple detector together. Block length is analyzed using carbon-13 NMR. Residual monomers can be analyzed using GCR NMR. Metal catalysts, these are used typically in the manufacturing process of polymer. The extent of contamination in the final raw material can be analyzed using ICP-MS or ICP-MS. N group chemistry is important for polymers, which can be assessed using NMR or a simple potentiometric technique. So glass transition temperature is analyzed using thermal techniques, which is DSE. And coming to the drug product characterization, I have listed some key characteristics here. So size distribution, laser diffraction is the primary technique which normally is used, and orthogonally SEM also can be used as a technique for assessing size distribution. And drug distribution is another important characteristics, where the cross-sectional Raman spectroscopy gives us understanding that how the drug is distributed inside the microsphere. And the next one is porosity, which is going to play a key role in how the drug is released on the from the surface, which can be assessed using mercury porosimeter. Trace components is important in microsphere manufacturing processes. There are some components are added in the process, which ultimately will go out of the product during the manufacturing process. So how much amount of trace components are left over after the manufacturing is assessed using NMR and LCMS, and it is important that these characteristics are comparable with reference product. Next is the most important drug release characteristics, which actually mimics how the drug is released over a period of time. And most useful technique is USP apparatus 4, and sometimes test tube water bath combination also can be used, and bottle rotating apparatus is also used in some of the different kind of microspheres. So next set of products is the characterization of peptides. So here I have listed different techniques which are most useful for characterization of peptides. So over a period of time, a lot of research has happened in this area. And recent FDA workshop also list very clearly what are the different techniques required for various characteristics. So most important thing to notice here is that for every characteristics which we study, so minimum 3 different techniques are used, 1 primary and 2 orthogonal. And isoelectric point is very important characteristics of the peptide, so which can be analyzed using capillary electrophoresis. Bioassay is another important key characteristics which are typically assessed using cell-based assays. Next important thing is characterization of iron nanoparticles, where the size distribution is analyzed using zeta sizer. Orthogonally, cryo-TEM is the most useful technique. Molecular weight distribution, traditionally a GPC method is used, and Mal's detector can be used as a orthogonal. Low molecular weight complexes is an important characteristics for the iron nanoparticles, which can be used, which can be analyzed using dialysis specific molecular weight cutoffs using ultrafiltration technique. And whatever is dialyzed solution can be analyzed for that iron content using ICP-MS. Labile iron is another important characteristics which is assessed for safety assessment. And the 2 orthogonal techniques are listed in literature: iron chelation assay and bleomycin assay. Surface charge is assessed using zeta potential, where the zeta sizer is used as a most useful technique. Iron core and morphology, microscopy is the most useful technique here. So AFM is the primary and TEM can be used as orthogonal. Crystallinity of the iron core can be assessed using XRD. In some cases, if the signals are very weak, synchrotron also can be used. And orthogonally, same kind of information and orthogonal information can be drawn from Raman, Temsed and Janes. The next important characteristics is iron environment, which can be assessed using mass absorption spectroscopy. Orthogonally, we can get the information using UV visible spectroscopy and ESR and Raman. Redox potential is assessed using polarography, and also orthogonally serimetric titration is used. Magnetic properties, 2 different techniques are most useful, VSM and SQUID. Carbohydrate shell characterization can be analyzed using FTIR and NMR, and in some cases size exclusion chromatography also can be used to assess bound and unbound characteristics. The next category of products is ophthalmic suspensions. Here, due to suspension nature, so drug is distributed in terms of particles. So size distribution is measured using laser diffraction, and orthogonally flow chem technique can be used. Morphology is assessed using microscopic techniques. So the 2 orthogonal techniques are listed here, SAM and morphology G3. Crystallinity can be assessed using XRD. So orthogonally Raman spectroscopy can be used. Most important parameter for the ophthalmics is rheology profile. So a rheometer which deals with, we can measure the viscosity at different shear rates. So rheology profile can be one of the important tool to assess the similarity of the reference product with generic product. And once the profile is established, a simple viscometer also can be used as a QC release test. So surface tension is important for the ophthalmic products. So simple techniques like tensiometer can be used. Drop size and drop assay is important, where the simple traditional LC assay, HPLC is a useful technique. In vitro release characteristics are measured. Most useful techniques are flow-through apparatus and bottle rotating apparatus. Polymer equivalence is assessed using SECMALS. So next section deals with the rationale for sampling and batch-to-batch variability for various types of complex products. So first important thing when we are dealing with the complex product characterization is we need to develop method which are suitable to characterize the key characteristics. And once the method is developed, the method variability is assessed by assessing the replicate analysis, and where the standard deviation is typically a measure of method variability. So once the method variability is understood, so using the developed method, we have to analyze multiple lots of RLD to assess the batch-to-batch variability of the reference product. And number of lots that is needed to represent the reference product variability is very important. And hence multiple lots of RLD analysis is very important to generate the range for acceptance for a generic product. And hence establishing equivalence criteria is useful for the characterization. And once the equivalence criteria is established, then the next step is the setting of the specification. So next I have listed different statistical approaches for comparative assessment. The first one is simplistic approach, minimum maximum range of multiple lots of RLD. And typically analytical method variability can be added to the minimum maximum range. Typically 1 SD is added to the minimum and maximum, and then range is established. The second approach is X sigma. So 3 sigma is the most useful technique, where the population of data is covered up to 99.7. So third approach is the tolerance interval. So typically 95% confidence interval is calculated, and which covers the 95 to 99% of the population of the data. And the fourth one is simple test, which is equivalence of test for means. Typical is the t-test. And the last one is multivariate analysis, also can be used as a statistical approach for comparative assessment. So here I have taken one of the examples for comparative assessment using X sigma. So this curve listed here is taken from one of the publications. So this graphical representation talks about 3 different characteristics of molecular weight distribution. And as you can see, the distribution can be measured using multiple sigmas depending on the type of population which we wanted to cover. This is one of the approach. And the next approach is the tolerance interval. So if the test results are variable, then the tolerance interval of the data can be used. So tolerance interval is typically a statistical interval with some confidence level, a specified proportion of the sample population false. So typically 95% confidence interval and 99% confidence intervals are calculated. And depending on the type of product what we are assessing, the rationale for sample size is selected. Typically a risk-based approach is taken depending on what kind of characteristics we are measuring. So the most useful technique is 95% confidence interval covering 99% population. And this technique uses the decision making based on coverage of population and method uncertainty. So here I have taken another example for equivalence test of means, which is using t-test. As this the graph represents comparison of molecular weight, average molecular weight of reference product and test product. As you can see from graph, the minimum and maximum values of test and reference is slightly different. However, that t-test calculation shows that the p-value is more than 0.05, and which shows that statistically there is no difference between means of both generic and reference product. So this is another example I have taken for multivariate analysis. I have taken it from recent publication, so where the NMR spectra is used for characterization. So multivariate analysis is also one of the technique. So where PCA scores are plotted, and then similarity of scores using Mahalanobis distance using a chemometric approach is used for comparison of reference products and test product. Thank you all. Thanks for the opportunity to speak in this occasion.
Great. Thank you so much, Maleswara. I'm going to now bring everybody back for the final panel. And this panel will address both the talk given by Dr. Reddy and also some of the questions, the fine questions that we're getting now in our chat box. So thank you. Keep them coming. Keep the questions coming. Folks, if you're on, go ahead and join the video. Hello, everybody, again. You can see the same familiar faces with slightly different order. It's like musical chairs, like Darby is now on this side of the aisle. But but you can see everybody smiling. We've all had our afternoon. Those last talks. That last talk was fantastic, wasn't it, everybody? Round of applause for Malaswara for bringing up all the data. And I noticed that he pulled data from some of the citizens' petitions where that's public information, guys. So that's valid to bring up. Thank you so much. So in the questions that we're getting some questions that lend themselves very specifically to control correspondences with the agency. So if you have questions about a product, a specific product, and I know you're trying to generalize those products, but the answers to some of those questions are it depends. And so you'll actually have, we recommend that you do send those in as controlled correspondences to us. For example, how do analytical characterizations differ for a specific product regarding submission to a 505B2 versus a 505J? I think that really depends on what the product is, and we will do our best to try to help you navigate the 505J versus 505B2 issue that was brought up earlier, both in the panel discussion with Dr. Leinberger and in the context of some of the other talks as well. I see also some questions about, let's see what else we have questions about. We have a question about variability for Q3. And I guess this gets at the sensitivity of the assay method, the reproducibility of the assay method, and the variability in the test product or your product, your batch-to-batch variation, etc. So what kind of variability is acceptable for Q3? I think this is a quality issue. So Pahala, is that something you want to respond to? And then maybe some industry folks to talk about how you control batch-to-batch variation for your test products as you develop them. So Pahala, go ahead.
Sure, Mark. In general, I'm talking about in general. In general, when you do you test the multiple lots of the reference products, right? And you got you got numbers, and you got the range, and you got the mean and the standard deviation, and then now you have the guide to where you need to have your the property of interest to be right. So now it's in general it should be within this variability of the RLD of the reference products numbers.
So you're saying that the test product should have no more variance than the reference product. Like if the reference product has wide variance, then that allows for greater permissibility for the test product, in essence.
Correct.
Is that a correct statement? Great. And so FDA sort of makes that determination from our information, and so some of that is proprietary information to the reference product, correct? But we do keep that in mind when we're looking at your information that you submit for your test products. So on to our industry colleagues. How do you help ensure lack of consistency in your batch-to-batches and in your analytical methods for those batches? Because the variance could come from both the measurement and what you're measuring, right? So Kevin or Dama, do you want to chime in, or Ravi?
Sure, Markham. Am I audible, Markham?
I can hear you. Yes, go ahead.
Thank you. So I think as Pahala rightly mentioned, so first thing we'll look at it, we'll look for as many as number of RED lots to look at the variability, and that we will consider obviously when we develop any method for Q3 or Q2, and it is important to have it so as minimum as possible the analytical variability. So that is what we look at it for any analytical method. And having said that, this is the first aspect we look at it. Look at the most of the RLD lots. What is the range we get it, And so first we'll ensure that the test product data to fall within that range with an analytical variability. And for some reason, so any of the test product, if it is going slightly beyond the RLD range, so the second important aspect to look at it is like what is the clinical relevance of that particular parameter going beyond the RLD range. If you are able to justify the clinical relevance and then show that as long as there is no impact, I think so agency is going to consider, where we have some specific examples. So that is the general approach we take it, particularly in this case where slightly we are away when compared with RLD.
Ravi, did you want to chime in there? Any thoughts, or Kevin or Rosario?
Yeah, so Markham, I agree with the Dharma's observations and statement, right, that yes, the recommend to evaluate multiple lots of RLDs and at least try to prove it that if we are going above the or range of the RLDs, how those characterizations or particular tests are related to the clinical significance of that. But more, if you are going above the RLD range, that means I think we all, at least I would strongly encourage team to look at it, why those differences are coming in. There must be some sort of, even if it is Q1, Q2, those differences may arise because of your difference in the manufacturing process or grade of excipients and all of those. So those evaluation needs to be done in proper orders.
Great.
And this is Rosario. Just to add on to what Ravi was saying, right? So it's really understanding that source of the variability. As we all know, right, the total variance is related to the variance from the analytical method itself and from the manufacturing process, right? You need to separate out the two. And then from that, you can need to dig deeper in the manufacturing process, which unit operation is causing that variability, or maybe it's a synergistic effect and you need to have the right control strategy. On the other hand, you have an analytical method. Is it the measurement itself that causing the variability, or is it the sample preparation causing the variability? So I think the aim is actually to reduce the variability and obviously to be within that of the RLD at the end of the day. Hopefully that provided some additional insights. Thank you.
Very nice. We have a question. I think someone in the audience is trying to stump us because they ask, what happens if the RLD is not available to analyze and all you have is a reference standard? And so this is a question that we get a lot, and it usually comes in the form of a control correspondence where someone says, oh, I'm working on this specific product, but the RLD is now no longer available on the market and it's withdrawn, and we may have some product and it's 10, 20 years old or something like that, and we don't have a good reference listed product to test to. But there is, FDA in its wisdom has promoted in the Orange Book a reference standard. What kind of Q1, Q2 should I be looking at for my test product? Darby, this is a common question that we get. Are you up for answering or giving a crack to answering this? You might unmute.
Yeah, I'm here. Sorry, this is Darby. Yeah, I think that there's always inherent challenges with that. I think really the aim of today's sort of talk is really to what kind of research we can do in that sort of space. Is it we need to do research or kind of come through aspects of how to better equate the RS, the RLD, or to come up with new approaches in that? I think to me, I see the general sort of question, but for today's focus, I'd really like to understand is where would we think they need the best research? Is it assessing those properties together? Is it using that reference standard? Because the reference standard essentially serves as the product that is the standard to test against. So that way, when an RLD becomes discontinued, you can still perform those comparative sort of testings and then be able to make those assessments. I do see the challenges that arise, but a lot of those specifics can come through as control correspondence. One thing I wanted to kind of present to...
Oh, sorry, Darby. If I may also chime in, that is an appropriate question for control correspondence. So the person in the audience that wanted, if that is a real problem for you, please do send it as a control correspondence. Some reference products are withdrawn for safety reasons, and that then becomes a concern, and we factor that in. Go ahead, Darby. You had something else to say.
Yeah, no, I'm just kind of following on both from that and also a couple conversations and tidbits I've heard, in particular about demonstrating sameness. The challenge is getting enough RLD batches to get this, 3, so you get an average and a standard deviation within range. So does industry and the public in general see the biggest sort of challenge a specific sameness assessment, like the best way in which to compare these two things together? Is this a challenge that we need to do new statistical approaches, research on that? Or is it in particular a specific sort of physical chemical property, a Q3 property? There's always an inherent challenge that we need to put more research into. In those instances there, it could be something like a rheological profile or even active ingredient sameness for complex mixtures. Do you mean? So I kind of put that to industry to kind of present what is the biggest challenge you feel that we could do research on that'd be a general tool for you guys to be able to utilize.
I mean, it's a difficult question to answer, really. It varies per product, whether it's statistical analysis, IVRR, all the stuff that we've been mentioning are all equal problems. To come up with one or two as the most important would target one or two products. We're talking about such a range of dosage forms here, such a range of issues that it's the broad brush research that you guys are doing across all of those dosage forms that we appreciate, and I think getting a two-way dialogue on those individual publications, those individual research, trying to understand how well the FDA view those papers and the output of those research, and how industry then wants to take them on, whether they agree with them or not, whether the industry understands or thinks of better or different ways of doing things. That conversation is where I think we can build on the research from GDUFA as it goes forward.
Great. Thank you. That sounded like a reasonable answer. Darby, you asked a tough question there. Did anyone else want to respond to that question? So the next question I have deals with economics, and this is the cost of technology that we're asking to implement, or you're implementing. I saw that Rosario mentioned some novel technologies, and some of that looked pretty expensive, Rosario. Would you agree? I know some of the new mass...
Yes, Markham.
Some of the new mass spec equipment, you mentioned maybe that's like $2 million each or something like that. I guess you can rent time on them, or have some other way to access those through an academic collaboration. But what are your thoughts on adaptation of new technologies that are pretty expensive to do, and what kinds of limitations do you put on? Would you put on that, and how do you get around some of that from an industry perspective?
Right. So I mean, I can speak to that. I'm not going to speak to the specifics of the cost instruments, but it's really around economics of scale, right? So if you're working on 3 or 4 complex products, at the end of the day, does it justify the need for purchasing the instrument? Because then you have to get it, qualify it, and then have the right person to run it and interpret the data. Or do you interface with a qualified CRO and then at the same time it right to get your analysis done? So it's a balancing act at the end of the day. Now, if it's going to be a technique that you're going to be using time and time again, where you're using it for characterization as well as potentially for release and stability, I give one example, like size exclusion chromatography, where you might be doing SEC-UV mols on characterization, but then you use SEC-UV for release and stability. That might be something which might make sense to bring in-house. But for something like AUC or SACS, or even more challenging SANS, that's something maybe where a CRO might be more appropriate.
Excellent, excellent. Thank you for that response. Does anyone else want to respond to that question? Kevin, you're at a company that has pretty good resources, yes?
Yes, we do, but I think the challenge is the same. It's the time to bring equipment in and qualify it and have somebody run it is only works when you've got the economy of scale that Rosario was mentioning. I think the difference that we struggle with a little bit, and I guess the industry does, is finding CROs with the right GMP status or GXP status to run some of these work that we're comfortable from a quality perspective presenting the data to the FDA and the other agencies, because we take that aspect of our work seriously, and it does preclude us from doing quite a bit of this work in universities that may not have that GXP status. So we are then going to look for CROs with the equipment rather than pure research organizations if we need to, and that's the other thing that we can struggle with, I guess.
I just want to pull it from the agency perspective. When we're looking at research and rewarding contracts, we are trying to, as much as possible, award to entities that would have the ability to widely propagate the availability of the technology, and also to have that technology available from more than one provider as much as possible, especially for new technologies that have very few people developing it. So we're very mindful of that. Of course, that does require having access to enough research funds to be able to collaborate with more than one researcher. And so that is a limitation somewhat on our part, but we do our best to identify all of the researchers in the field and to have as competitive and as level a playing field for that research as possible. I just wanted to put it out there for the public to know how we look at prioritizing research and our research partners to develop these analytical technologies. So, Darby, I saw there was a question posed for you specifically. What aspects of structure and impurity testing are critical in the API sameness discussion, and how can this be demonstrated? And they're referring to slide 8 of your presentation. I don't know if we can project that.
I think probably Ram and Pahala would be best positioned to discuss that, especially about API sameness, because I know this is much more in their wheelhouse. I presented research and data from a lot of different groups, so I'll hand over to the experts.
Ram or Pahala, Darby is punting the question over to you. He's asking for a lifeline here. Do you want to be a millionaire?
The API, Ram is the API guru, so I let Ram.
All right, Ram, you have been pointed out.
All right, yeah. So let me say, I think the Madheshwara's talk was particularly very, very good, where he pointed out that for the different kind of products, all the different kind of instrumentation one would use. I think what I want to say is what he pointed out is that it is not one particular instrument, it is the totality of the approach and the number of techniques that may be needed to get to the answer. So when we talk about the API and the API impurities, it gets back to what Rosario started with by saying that we need to have a reverse engineering. So reverse engineering is trying to understand the drug substance or the drug product or the formulation, and I think the question is about the API. So trying to understand the drug substance and then the impurities are you saying those are impurities or those are concomitant compounds? And I think this is an important distinction that researcher or the industry has to address. And this distinction will come from understanding of the RLD or of the API. If you are looking at conjugated estrogen, and then you will have, let's say, 160, 170, 180 steroidal components. Understanding of those steroidal components or the composition of the API, and it could be any number. I just gave that as an example. And then the other point which was brought up about the variability. So one is understanding RLD, and second is understanding batch-to-batch variability. It could be batch-to-batch variability, year-to-year variability, season-to-season variability, understanding the variability. And I think a progress, and the question was asked about the area where we might need a progress, and that may be a statistical analysis of all the components of the RLD and coming up with an algorithm to define what is pharmaceutical equivalence. And going back to Darby's talk, where he talked about how FDA is, he gave example in each one of these cases, whether it is an API or excipient or bioequivalence or formulation, and how agency is keeping up with the current technology and how they're trying to not only present it to the outside world, but also training the reviewers within the agency. So these collective efforts and understanding the need of different analytical methods, it is not just one, but number of analytical methods, the product and the complexity or the variability, reason for the complex, and manufacturing process is essential to promote a complex
I would say
product.
each effort that we have, each lesson learned, like there are iterations to lessons learned as we go through time. We had, for example, the BPA issue come up with container closures systems, right, for plastics leaching BPA. Do you remember that? That was like 10 years ago. And we had issues about nanoparticles showing up in product. And now we had the nitrosamine issue, as was mentioned during the open public session by 3 of the speakers there. And the agency has learned from each of those and applied those, some of what we learned towards making sure that we have sufficient assurance of safety of the ingredients, including the API. Would you characterize that as accurate, Ram?
Markham, I think very well put. Maybe I just want to add one more thing, and probably maybe we can have a discussion on that. And I think it was Dama or somebody brought up a clinical relevance. And I think this is a difficult topic to discuss in a generic setting as an ANDA under the Waxman-Hatch amendment. And I think if somebody has more thoughts, I think we'll be glad to hear.
All right. I'm going to again encourage folks in the audience. If you have interesting questions for us, or even not so interesting questions, we'll take those as well. But as long as they're relevant to the topic at hand, please type them into the comments box, and we're happy to address those. And we have folks standing by to look at those. Yes. Who's that?
Yeah, Mark, while we get, this is Rosario, while we get more questions from the audience, just to add on to Ram's point about API and API sameness, I mean, really, it requires good detective work, right? When you're doing the reverse engineering, you need to ensure that you're also not changing the API itself. So when you're doing your comparability, you're comparing, apple to apple. Moreover, you also need to ensure, during your manufacturing process, you're not changing the API itself, right? That's why it's really important to compare the API to the final product. But in some cases, the API does change, right? And that's fine, right? Because it also changed for the RLD. So making those logical connections and really understanding the API sameness from those perspectives and actually explaining it and developing that storyline is super important for product development.
Okay, I have a question here. First of all, the question is about outside partners for FDA and industry. And I saw a prior question before Maleswara's talk as well about how USP can be involved. So I'm going to suggest stakeholders and other standards organizations. How can they get involved in the analytical space here? What sort of functions they are performing currently, in your opinion? And also, what kind of functions can they perform in the future? Or is there room to expand on those functions? We all know about USP apparatuses, right? For dissolution and other things. Maybe some of you folks might have some thoughts about that. If you have bad thoughts, hold those to yourselves, or you can coach them in a politically appropriate way here in the discussion. Please go ahead. Who wants to go first? Rosario?
Markham, this is
Maleswara. Yes, go ahead.
Yeah. So, Markham, I think the best way is to the FDA, when they are sponsoring the projects, go to a best professor in a renowned university and ask him to do the research. He's the best person who is most knowledgeable, most senior people available in different universities, and that's the best way to start with. And that is available for a public publication, so that everybody will get benefited from that.
I think it's the accessibility of the research too. For example, if there's funded research in a university, that should be published as much as possible. If it's USP apparatuses that are called for use in a standard dissolution process, that apparatus should be widely available for everyone to use. Oh, are you folks on, Kevin? I'm seeing some frozen faces. Oh,
yeah,
good.
We're
okay.
all on, Markham. So maybe, Markham, there's a couple of questions coming in. I mean, there's a good question there, and maybe the FDA can take this one. Should all the analytical testing be conducted under GMPs for submission characterization? Maybe you can shed some light around that, your thoughts, perspective. Thank you.
So GMP requirements for analytical testing. I'm going to have Pahala and Darby address those first. I think it depends on what kind of testing is being done and whether GMP is required or not. Darby, do you want to take it from the BE perspective regarding analytical testing and what the requirements are for GMP, and then Pahala maybe on the quality perspective. Go ahead, Darby.
I believe for the one-time study, especially like the analytical methods that you've been discussing, the more complex ones, SANS and SACs and things like that, where you'd have to do through a very specific, government or specific research organization, those don't necessarily have to be GMP. They just you need to obviously have the methods shown well and robust, and some of those sort of additional sort of supportive information. But like I said, so I think for those types of analytical methods, there's a little bit more sort of aspect to it. I don't have a full depth. I think I'd really kind of defer to Pahala on a little bit more on the review sort of aspect of those.
Darby, you are on spot. I guess to make it easy, one-time study versus routine testing. Those kind of like the two buckets, right? One time versus something that needs to be under GMP is for the those testing that are routinely done for the drug products.
Okay. Was that and Rosario? Did you want to comment on that as well? Do you conduct
No,
all your analytical testing under GMP?
I mean that was the answer. It definitely was clarifying what I was asking, and I think to Darby's point, right, like SAC, SANS, these more complicated instruments, it's going to be very hard to have them as GMP instruments. But good laboratory practices need to be used, right? They can be robust, making sure that all the data is recorded, and there's the right data integrity there. So I think from that's a good answer. And then the more routine ones, obviously like SEC or SEC mols or SEC UV mol, those could be potentially routine, right? So maybe the SEC UV, that in that case, that my understanding would need to be validated and under GMP. Correct, Ram?
The methods that you would use for release and stability, they will be GMP, one-time characterization in-depth study, and I think as everybody pointed out, Paula and Darby and Rosario, you good documentation, yeah.
We have a question about population bioequivalence showing sameness. I think this is more of a BE, almost a modeling type of question or a pop PK type of question. So population bioequivalence as a demonstration of sameness. There are some analytical tools used in population pharmacokinetics. Maybe we can expand on this question a little bit more. And so things like blood spot chemistry and things like that. Do folks have experience using that, and what kind of hurdles do we encounter regarding analytical methods using those kinds of techniques? Does anyone want to chime in there?
Actually be referring to the statistical approach of population bioequivalence, which is different.
Right, but I'm going to pull it back to the analytical methods, because the statistical piece we're not going to be talking about here in this session. But I think there are analytical issues when it comes to looking at population bioequivalence, especially if you go out and you're sampling from the real world. And so this gets at blood spot PKs and things like that. I think there are issues. Maybe I can start answering that, but I want to hear from some of the others as well. There are some issues around stability of the analytes, for example, in the blood spot. What is your reference? Are you going to be looking at hemoglobin in the blood spot as your sample reference for amount of drug? There are some challenges to doing that. Of course, some of that has been worked out. Does anyone here have experience doing this, or with analytical chemistry techniques using those kinds of techniques? I guess not. Okay, we'll skip over this question then. I got the next question. Can you please address how to analyze transdermal products using new technologies? Transdermal. So transdermal, there are two types of transdermal products. One are those with backing materials, like the transdermal systems with patch-like technologies. And then there are topical products like nitroglycerin gel, for example, or lidocaine gel, where you have a transdermal product, and you're putting the substance on the skin to get blood levels that then go to the site of action. I guess the thought is, how do you analyze those products using new technology here? Is anyone developing transdermal products? Pahala? We have some of those products in our wheelhouse.
Yeah, I'm not really too sure about the questions, whether, like what you said, Markham, is it patches related or the conventional cream gel lotion type transdermal product?
The answer.
And then in terms of the tech,
I was going to say that the answer depends on those technologies, but we're getting, because of the systemic action of those transdermal products, we're going to be measuring blood levels. So you basically rely on blood level technology. And I don't know about new technology. A lot of that is like the lower limit of detection, right? What's available for some of those products. And a lot of the products we have over the last decade been lowering the lower level detection for those.
I mean, one of the commonly used, if it's like measuring the API in the bloodstream, it could be LCMS. Mm-hmm. I think that one is quite sensitive.
Great. And so I think we have several more minutes. It says here 45 more minutes. Andy, any more questions do you have questions for each other that you might want to ask?
If I may ask folks from panels, from industry, do you guys have any comments or any thoughts?
I'll throw a question out that might get another conversation started. I mean, artificial intelligence and people moving through that side of the sort of world is obviously taking on where the future's going. Where does the panel see that affecting the analytical techniques and the analysis that we're moving towards in the next 5 years or so?
I'll take a bit on that one, Kevin. I think it's quite good. I think a lot of it's going to become how you're going to actually analyze complex data. We've seen an awful lot with like images, where you're able to do cross-sectional imaging. I think I showed in one graph PLGA microspheres with drug distribution and porosity. Now it's a lot of image analysis and resolution. So AI can then be taught kind of to do some of that imaging recognition, similar to like facials being already used, but now being able to teach it to kind of identify specific sort of regions of drug versus porosity, and then do an equation of that to then do a comparison. So now you're gonna be able to do image comparisons on a much larger level, which would be prohibitive in the past. I think one thing that always with microscopy and other images has always been just the ways in which we can compare. It's always been very rudimentary. Computers have kind of made a next sort of shift, so we can start doing particle size characterization just on an outline. Now I think it's going to go to that next level, and we're going to be able to feed in lots and lots of images to be able to get a lot more information in that. And I think ultimately what I could see as the benefit is that you'll be able to start to see small manufacturing differences or small aspects that we might not necessarily be able to identify on a human eye or small sort of selection basis, being able to identify those and then being able to correlate that potentially to a pharmacokinetic or an ultimate PD sort of endpoint or bioequivalence component. So in essence, kind of bridging those two components together. That to me is an exciting new realm to kind of start to dive into, where I think AI is going to really start to push. But that's just my analytical background and looking at those. I definitely recommend anybody else who wants to jump in on that.
Yes. So Kevin and Darby, interesting point, right? The DGM technology is very interesting. However, that could be a whole PhD research in itself, right? You're correlating, the porosity, the inherent viscosity, and other microstructure to now release profiles, right? And what do those actually changes mean to the release profiles, and then building a model. Then what does that mean, from in a bioequivalence point of view as well, the next level. So I think it's, interesting. The question is that it is a huge body of work, and who will actually do all of that work at the end of the day.
Coming out, but I think it's still the bit that would be interested in is how we bridge that gap to the clinical relevance, the PK modeling, how similar or any minor differences that these techniques pick up. How do we potentially use AI and real clinical data to really get to the understanding the variability that's allowed or acceptable in products and what that has, and where we can feed that back into the sort of the generics industry in terms of similarity. So I think there's a huge body of work that could come out there.
Is AI being used at identifying spectroscopic signals and things like that? Maybe machine learning for as far as curve signatures and stuff like that?
Yes, Markham. This is Malleshwar. AI being is widely used now. We are already experimenting using AI for image analysis, for spectroscopic analysis, even for the chromatography, peak assignments, and chromatographic method development. So various areas the AI is coming up now.
Excellent.
I mean, to add to that, AI is being used for years, right? When PAT first came out, and you're doing chemical imaging, and you're looking inside tablets and things of that nature. Raman confocal imaging and NIR chemical imaging. So it's been used, right? And I think it's getting a refresh now. I think you mentioned the chromatography. It's been used for peak tracking and other things as well. The question is how can, and now we're getting even more data. How can we combine the elements or information from multiple techniques, right, to the in vivo data? And is it relevant at the end, right? That's the biggest question that we have to ask ourselves. Will it be relevant? So we could have all these minor, let's say, differences, but are they relevant? But sometimes the minor differences could be relevant.
So I wanted to go back to the USP question. So there are standards organizations. There's the European Formulary, there's the USP. I think there's a role to play of those organizations to standardize like what are acceptable for exchangeable excipients. Like, can you use this excipient in place of another excipient? And what are the parameters around certain excipients? For example, white petrolatum. How do you distinguish one white petrolatum for another, and which are acceptable for substitution, or other molecules, other components that are used in the kind of water, even, for example. These are all things that USP and other standards organizations can help devise, like, oh, this is what a clean spectrum looks like when you use this analytical tool to look at this excipient, and if you use another excipient that has the same parameters, then that's acceptable for use. It gets at what is Q1? What do we allow for Q1 in that context?
Yeah, Markham, again, Malleshwar here. So we actually extensively work with USP for various activities. Some of the very advanced techniques are available in USP labs, but they have some internal difficulties in sharing this particular kind of equipments for industry use, especially when it comes to data to be submitted to agency. For research, they will allow, but they have difficulties to, when we ask them permission that can we go ahead and submit with your FDA, so they said no.
So this is what I meant about some of the political sensitivities around this issue. I think it's important for this is something that maybe our USP folks in the audience could take back and think about, like what kind of data can be made publicly available for use to help further generic drug development in that context. We're all partners in this. Do you want to join us in that partnership? And it's something to think about. Does Kevin or Rosario want to say something about this, or Ravi, Dama, Maleswara?
Yeah, so Markham, I just wanted to add one more point. So one appreciation to FDA that FDA is significantly stepped up their engagement with the generic industry in terms of pre-development meetings, in terms of and mid-review cycle meetings and all those things. But one specific aspect which I have noticed is only when you come close to their expectation, they will start disclosing something which is really in detail, and not if you are a little further, they will just give you a broad deficiency that you are far away. So maybe it's kind of slow release pattern. There is a scope for improvement there, more engaging dialogue during review, and scope for discussion as Rosario is indicated in his presentation.
So you're suggesting a sort of red light, green light, or a pin the tail on the donkey type of approach to the queries about Q1, Q2. I'm not sure that's feasible in the context of information disclosure, but it's certainly an interesting suggestion. Darby, what are your thoughts on that? Oh.
This is not specific to Q1, Q2, Markham. I'm specifically talking about the areas where we can engage in a dialogue with the players in the industry to how to crack the complexity of complex generics much faster.
So you're saying for complex products to have a discussion with industry about other players in industry about what's the same or not, as opposed to the agency.
Yeah, so we need more dialogue in that area, because sometimes even at agency level there is a parallel research going on. So if we have a dialogue, together we can crack faster.
Well, you have other folks here in industry. What do you folks think about that? Is that dialogue worthwhile? Is that something that, how difficult is it to achieve that kind of dialogue, given the I think multiple folks, including the conversation with Rob, have suggested that there's proprietary information that's difficult to get out there.
But I do think there's huge difficulty in openly discussing between ourselves. These sorts of meetings, though, do bring us together and help us to open those channels. So it's coming to these sort of industry conferences, workshops, and especially when they're held like this, that would open and start that conversation. So it's difficult. It's never going to be easy, but at the end of the day, we are all scientists and with the same goal. We just work for different companies and have to be respectful of each other's.
I would also add that there's structural difficulties for generic companies to talk with each other. The issues being, as some of you have seen, about anti-competitive behavior, collaborating with others on price fixing, things like that. So how industry talks with each other is something that you all I can see the hurdles to doing that. If I were to give a talk on how industry talks with each other, you have a whole slide with lists about why you have difficulty talking with each other, right? And then what could be the things that you can gain from talking with each other, and then you want to think about, oh well, what are the best venues for you to talk with each other in an allowable manner, and what sort of topics can you talk about without crossing the barrier, going into an area that you're prohibited from talking with each other on.
So Markham, I mean, here's a thought, right? So you have an independent, right, third party, you have CRCG. Maybe there's some analog compounds that could be used in certain complex cases, and kind of a workshop or lab that would be built around it. And then, for example, that workshop could be around sample preparation for X, Y, and Z using multiple techniques. And it could be an analog compound, but in a specific product class. So maybe, it'd be interesting to entertain an idea around that.
So using the center as a venue for discussion about scientific issues only, I think that's a reasonable thought. And that's something that maybe we can talk to Jim Poley and others there about. In advancing generic industry science as a whole. Science is not I think science is a solitary activity, like the Isaac Newtons of the world, but then there's science in a collaborative manner, and you'll see like all the Nobel Prizes recently are all collaborative efforts. There are multiple scientists involved, and so the best science currently comes from multiple labs collaborating with each other, moving those things forward.
Yeah, and Mark, I fully agree. Really advancing science to pioneer access for patients. I think that's ultimately our goal.
Yes. Well, that's excellent. Likan says there's a long question in the comment section. Let me see if I can pull this up. So there's a question about sharing information. Oh, I guess we've sort of discussed this about industry sharing information in the context of working together, and this one specifically about in the combination product space. I think, and we heard a number of folks talk, and in fact in Rob's session there was a long discussion about human factors and how that's a major hurdle to moving forward in the combination drug device product area. This is not really an analytical methods kind of question, although you can is human factors an analytical type of effort. There, I guess you can say that it's sort of a psychometric analytical method. If psychoanalysis is an analytical method, but I think that's pushing it, right? But at the same time, sharing information in that space could be useful. I think that's the comment there, and using the structure of the center for complex generics as a method. There's a question about the Q1, Q2 process being more transparent. If there are several ingredients, how can someone respond? Can we respond to Q1, Q2 about where they have met the Q1, Q2, as opposed to the standard response where we just say no, you have not met the hurdle. I think there's disclosure issues, and the disclosure being proprietary information, and how does the agency, what is the cutoff for disclosure? Darby, do you want to address this, because you've handled a number of Q1, Q2 queries, right, in your time? Darby?
Yeah, Mark, what was the, I mean, these are very specific questions about like more regulatory sort of policy and things like that. I think there's definitely a need to have those types of conversations and kind of the feedback to the control correspondence route. I think for like I said, for this session here, one of the things we'd really want to know is what scientific research can we do to benefit that. I think that's a little bit different to, it's a good question. It's definitely a valid question. I just think it's the core aim here. We need to figure out what our research priorities will be for the next fiscal year. If it can be within Q1, Q2, my comment there would be like, what methods can we use, similar to like what we've been doing to characterize the, polymeric sameness and things like that to demonstrate comparative this and there, and to support Q1 sameness. There's obviously always the challenge back and forth about the regs versus the science that's going forward, and being able to get those to marry up and provide the justification, how science can then help provide the justification that this product is safe and effective and meets the other Q1, Q2 requirements from the regulations.
I think
if you wouldn't mind, Markham, I would like to ask
Sure.
industry another kind of question specifically, especially on the liposome products, because in the last couple of years, we've been able to approve, now I think it's 3 liposomal doxorubicins. What does industry still see as the inherent challenge with liposomal products, especially about the analytical methods to characterize it that we need? Do we need to do research on the methods to characterize it? Is it methods for bioanalytics to get that better? Is it methods or just recipes and standards? What does industry see as the next push or need that we need to do in liposome products to get that rest of those products, generics, developed and available?
Yeah, so this is Malleshwar. I don't see a big challenge in availability of information about how to characterize liposomes, but there are key challenge here is to meet the sameness through process design. I think there is enough publications and literature and FDA information about what are the kind of tests which we need to perform, and what kind of methods we work, and then some of the publications are done by innovators themselves. But the key challenge is to really meet the sameness using all the orthogonal techniques. If you are listing down 15 different characteristics, so matching 13 is not okay. So you need to match 15. So if 1 or 2 is really not matching, so it is not, sameness is not achieved. So how much is the process design makes sameness closer to the reference product? That's key challenge. Of course, bioanalytical challenge definitely exists, which I'm not an expert, but characterization is largely evolved for liposomes.
This is the deformulation issue of you can measure what's happened at the end product, but you also have to step back to what actually went into the product itself to make it. And so when you talk about here's Q1, Q2, you may be off because certain things have been removed during the manufacturing process, or things have been added and then removed. So then how much of that still remains at the end of the process?
Yes. So this is common to even microsphere products. So liposomes also in some places, yes. What is added and what is going out, and how much is the remaining components sameness, and how does it impact the release characteristics and in vivo characteristics?
Right.
So this might also get to do what Dama was also speaking about. Oh, go ahead.
Yeah, so Markham, as Dr. Reddy mentioned, so this is most of the complex projects today going to the public literature as well as, so the research available, the literature available. So majority of the products, like it could be, glycosaminoglycans or the polysaccharides or the microspheres or liposomes, the end product characterization is very clear. So majority of the people knows it, what is needed and what are the analytical techniques which are required for the characterization of these products to look at it. But interesting thing is, like, these products, majority of the products are product by process. So what are the process steps involved, and then at various stages, what are the identification of the right CQAs to control at intermediate stages in order to ensure the right sameness of the product. So that understanding need to be more developed from an industry perspective. I think so, where few of the companies are working towards that direction using few of the pad controls as well as the offline analytical techniques to understand at each of the process stages, to use to monitor the right quality attributes to end up with the final the sameness attributes. So that is what is most important for all these products. That's what I think. Was I audible, Markham?
Sorry, I was on mute. I need this thing like you are on mute. So we were just discussing that this is the reverse engineering type of utility for analytical methods when you're taking the reference product and you're doing those initial discovery of what is it made, and they're experts at these, and they're actually firms that just specialize in doing that. And there are a lot of little tricks that they use to figure out what's in the formulation, how it was put together, etc.
Yes.
Some of those are proprietary methods, I imagine. Yeah.
Yes. You are right, Malcolm.
But like what Abhi was saying, some of the products are much more difficult to get at, like the liposomal products. Whenever you have more than one phase of a product, that adds additional complexity to an already complex product. So I'm looking at some of the latest questions popping up. Let's see. So it sounds like a lot of our discussions today are about cooperation and sharing information with each other as we move along, both in the research environment, getting things published, making sure that if we're doing the research that we're doing, we're discussing doing the right research moving forward, and we do our best to make sure that the research is published in a timely manner. And then they get translated into informational needs for bioequivalence in the form of product-specific guidances and things like that. Some questions arise as to whether analytical methods should be mentioned in the product-specific guidances. I think it varies. It depends on what the product-specific guidance is. Does folks in industry have any thoughts about what you would like to see as far as detailing goes for analytical methods in the product-specific guidance? That's a catch-22 there, because if it's in the product-specific guidance, then people follow that, and then if you come up with a different method, that restricts your ability to use other methods. But at the same time, if you don't know what method to use, it can be helpful. So it's like a double-edged sword of including certain information in product-specific guidances. I'd like to hear from industry what they think of how much information should the agency be putting into product-specific guidances regarding analytical methods. So anyone in the industry on the panel?
Yeah, Malcolm, this is Malleshwar. I think the best approach is to publish the information of research as a public information rather than restricting as part of the guidance.
And which is what we do now, right? So we do our best to publish what we have and get it out. They also, some of them are coming up as posters. So you'll see, sometimes it's hard to publish on a specific analytical, as you know, the amount, how much it contributes to the scientific universe. Editors of certain journals will say, oh, that's not publishable. So we do present a lot of that as posters at meetings. But we do try to get as much of it in the public domain as possible. Any other thoughts on that, Rosario, Kevin, Dama?
Yeah, I kind of echo what was said before, right? I mean, we don't want to be too specific, right? At the same time, if something is mentioned in the guidance and something is known, for example, if you're doing X test, don't use a quartz cuvette, use a different type of cuvette. That could be helpful, right? If something is going to be listed in the guidance itself as a specific technique, if it's known already. So then you don't need to go and start investigating, different types of, cuvettes, for example, and that's one technique. Or like, for example, if you have another product and you know that XYZ is going to cause interference, right? Maybe a short note around that could be helpful in the guidance. Thank you.
Okay, we'll do our best to predict what kind of mistakes people can make.
Markham, I think as Dr. Rosario mentioned, definitely it would be helpful to publish some of the information, like with respect to extraction processes which involve to isolate the API from the active pharmaceutical ingredient from the complex formulations, because probably that could be one of the common approach industry can use it, and subsequently mentioning the technique in the product specific guidance. Of course, there are pros and cons. Rather restricting to a particular technique. So when there is availability of an alternative technique that can be used for various signatures or for the quantifications, and so definitely these wherever the isolation approaches, particularly for example, like it could be where the complex product like sevelamer carbonate or cholesevelam hydrochloride or sucralfate. So these are the few complex formulations where if the product specific guidance comes with the API extraction approach, basically that would definitely help industry to speed up the analytical activities.
Yeah. We have a question here from Anna Schwendeman about the main challenges in the iron sucrose approval. I'm not sure what we can say about that. Darby, do you want to say whether we can say anything about that?
I'd really like to hear from industry in terms of what are the challenges they've seen and faced. I know we've done a lot of GDUFA research and the analytics and also the bioanalytical methods for the iron carbohydrate drug products in general. So I think something we'd like to get feedback on is where do you feel that there's still a need? What's still the gap or the aspect that we need to continue to do more research into? I feel like this is one of the drug products we've done an awful lot already in. So anything
we
that we
published
can
some
do
of that
to facilitate that.
And Darby, we published some of that as well?
Oh, extensively, yes.
Great.
Yes.
So industry,
So,
what are your thoughts about iron sucrose? What challenges have you encountered in your development journey for iron sucrose?
yeah, I think Dama has touched upon this topic, Markham, that it is the process design. So how do you design the process to get the molecule which is same as reference product? I think FDA has published extensive articles using multiple techniques how to characterize.
Anyone else want to talk about iron sucrose?
If I may, making a plug, there will be one presentation in the upcoming SBIA workshop, which will take place in September. One of the talk on iron colloids, looking at it from quality perspective.
Great, and that's a public meeting. Anna, we hope that you can make it there and you can hear the talk on the iron sucrose. that Pahala was saying. Is that coming from the FDA, Pahala, that presentation?
Correct, yes.
Great. Let's see. Any other questions? So we're told we have 8 minutes left to go.
So, Markham,
go ahead. We can close up with final remarks, I guess, as we move along. And since there are about 8 of us, maybe each of us has 1 minute to say about what you thought about this session and what the takeaway points for you are from this session. Is that reasonable? Great. So I'm going to just go through the list, and maybe you can spend 1 minute saying your closing remarks for this session. And thank you, especially for me, just thanks to the audience for being such a great audience. All 200 or 300 of you. I think we have quite a number of audience here. So I'll start going down on the list. Pahala, do you want to say some closing remarks?
Cool conversation and discussion. All the presentations are very impressive. My takeaway is, it's a two-way street. We work together. We learn from each other. In fact, personally, the FDA, I learned a lot more from you guys. You guys have been good educators for me to, to understand, where we are coming from, what kind of technology, and then so on and so forth. And they kind of show it here, from all of the presentation and the input. So this is excellent, and I really enjoy it. Thank you.
Thank you, Paula. On my screen, I have Kevin next. Kevin.
For hosting the event, it's a really important opportunity for us to meet and build trust, and I think that trust is so critical in everything that we're doing, and for the audience and everybody else. Thank you to the presenters just for such good insights into what's going on. And for me, my takeaway is really, I think the bit that I've caught in my head is trying to work out the opportunity to open up some broader discussions. I think I've attended a few of these types of meetings, and the difficulty that I think the industry has talking openly amongst each other, that there should be ways of tackling that, because I think we've all got the similar problems, we've all got the similar interests, and we know that getting products to the patient is what we're all doing this for. So I think that's my takeaway. I'll have a good work and think, and maybe contact a few of the industry colleagues and see what we could come up with as well. But thank you to everybody. It is really interesting. So thank you.
Thank you, Kevin. And just from Darby and myself, we're if you need us for anything, we're open for a discussion. We keep an open door. So if any of you need to ask us anything, we're here. Rosario, you're next on my window here. Rosario?
I am here. It was not turning on for whatever reason. Okay, so definitely want to thank the host, the FDA, the audience, and the presenters, right, for a really, really great, engaging conversation. I learned a lot. I really appreciated the openness of the conversations. I think the frankness. I think that's really is going to lay the foreground for future conversations, right, in a more collaborative fashion, whatever that may look like. I think we heard something about cracking the complexity code. I think this is going to be one of the parts that could help to crack the complexity code. Along with other things, right? I think the research CRCG is doing and kind of providing that independent third-party assessment, maybe those kind of open labs where different companies are invited and they work on some type of analog compound could be helpful. And lastly, right, I think we've heard it loud and clear. It's increasing that openness interaction with the FDA, that dialogue, the control correspondence. Could that be an additional mechanism for getting faster insights into methods? And then the product development meetings. Could there be an even faster way of interfacing with the FDA to get acceptance of those meetings much earlier on without providing that big body content up front? And just to close, really, at the end of the day, all this great work to bring these complex GX to market really is to provide access to patients. So thank you.
Ravi? We're told we have 3 minutes left, so we have to speed it up a little bit.
Just to respect the times, right? Thank you very much from the industry to the FDAs and the presenters that are presenting the nice contents and the details providing those things. We're looking forward for more and more this kind of sessions for future. So, it can bring the industry and the agencies together for what kind of research that we're looking for futures and everything. Thank you, everyone.
Excellent. Excellent. Research is key. Ram?
At one point, I saw there were more than 225 participants in our session. That is very, very encouraging. It speaks to the point there is how much interest is there in the industry on this topic. And we really identified issues where FDA can help and also industry sharing, and that was a very, very wonderful start of the discussion. Thank you very much.
And Darby?
Everybody else, it's been a great conversation. I think it's really valuable both to, hopefully to industry, but I know it's definitely valuable to myself and to FDA probably in particular, to understand a bit better about some of the challenges, some of the areas in which we want to go, and where we can really start to push some of this research. I really encourage all industry to have those sort of frank discussions as much as possible. We all are kind of on the same scientific level. We all want to have the same sort of scientific discussions. I think we all enjoy it. And I think it's really to ultimately get to the ultimate aim, I think, as Kevin, you said, is to get high quality generics available to the public. So I think that we all have the same goal in mind.
Great. Thank you, Darby. Adama, and then Malaswara. It looks like we're wrapping up soon.
Yeah. Thanks to the organizers as well as thanks to the presenters. It's a great the opportunity to part of this workshop. And really enjoyed. I think this collaboration, industry collaboration with agency definitely will help to speed up the complex generics to bring into the needy of the patients. So it's a great opportunity to be part of this. Thank you very much.
Great. And Malaswara, you're the last on the panel.
Yeah, Malcolm. So thank you so much, Malcolm, for getting this workshop run so excitingly. And I learned a lot. So we will definitely engage more with FDA and increase the dialogue with FDA to crack the complex generics much faster.
Excellent, excellent. Thank you. Just for your point on RAM, we had a maximum, I counted, of like 252 participants at the peak. But folks had other things to do perhaps in the afternoon, but we're still left with close to 200 right now, almost 200 right now. So we're going to wrap up and we're going to go to the main session to hear a keynote closeout for the whole day, right? So on to Mark. Take us away over to the admin layout.